Okay, very good morning to all of you. So, myself, I'm Ravi, lecturer in zoology. Today we are going to discuss about in a human physiology, in a excretion and elimination concept. We have a core concept as the urine formation. In a mammals, how the urine is going to be formed, we are going to discuss it here. Okay. Then how it was going to be happen, let us see. Then see, before getting into the urine formation, we have to know few points about the structure of nephron. Okay, how the nephron it was made. Why? Because in humans, the entire kidney it is made up of the nephrons only. Because the nephron is a structural and functional unit of the kidney. And here, if we see, this is whatever the human nephron we have, approximately 3 centimeters long, and we have the cortical and as well juxtamedullary nephrons, as of we know. In that, we drawn it as a juxtamedullary nephron, and let us see this limb. And here it is an afferent atriole, and this part is an afferent. And as well it is a collecting duct, and as well here we have a ECT, and this one is a DCT, proximal convoluted tubule, and as well distal convoluted tubule. And the main points here, the loop of Henle. And as well, here we have, if you see clearly, here we have this Clomsed region. This one we call it as a glomerulus, and as well, this one we call it as a Bowman's capsule. And here, this combinedly, Bowman's capsule and as well glomerulus, combinedly we are calling as a Malpighian body. Okay. Then let us see here. These are the parts of nephron what we have. Okay. And in the nephron, these are the parts we have. Then now see how this part of the nephron it was helping us in the formation of urine. Okay. Then see from the starting. In a urine formation, we have the few steps. Let us start from the first step. This is ultrafiltration. In ultrafiltration, what is ultrafiltration here? Okay. In an ultrafiltration process, it happens here. Okay. Ultrafiltration happens in this Bowman's, this Malpighian body, and in the Malpighian body we have the Bowman's capsule and glomerulus. And see from the start. Here we have the afferent arteriole. Let us, for our convenience, draw this out. We have this part, afferent arteriole, which is a wider. Afferent arteriole, which is a wider. As of we know, if it's a wider, definitely it can able to carry the blood, and along with blood cells, nutrients and all. Okay, all was carried by this, and now this one is carrying in this direction, and then here it formed a bunch of blood vessels. That is what we call it as a Bowman. That is what we call it as a glomerulus. And then from here there is a blood vessel it was arising. That is what we call it as a afferent arteriole. And the thing here we have to remember, here is all the portion it was happening in this capsule. And see this. Here, this is an area. Okay, about this area we are going to discuss what ultrafiltration it was happening here. Whenever the blood came here in this direction, at that time, the blood comes with a respective, the pressure. That pressure is a 60 mmHg. With this pressure, the blood usually comes in. When the blood came here, at that time, here, already it have the somewhat blood vessels. Okay. This blood vessels, it have the plasma, and that plasma, as of we know, we That's why it maintains the somewhat osmotic pressure. Osmotic pressure, and this osmotic pressure exerted by plasma. It is a 10 mm, 30 mm Hg. And here it is a 30 mm Hg, and here it is a 60 mm Hg. With 60 mm Hg the blood came, and this 30 mm Hg giving an opposite action to this blood pressure. Why? Because if that same pressure the blood enters here, this area can't able to be tolerated. So that is the reason it was giving an opposite action. Now see here, we already know here there is an interstitial fluid. This interstitial fluid, it have the pressure it accepts 10 mm Hg again, okay, and this is also gives opposite action to this plate. And as well here there is a nephric filtrate, whatever it was filtered, that nephric filtrate, it also contributes a somewhat pressure, that is also 10 mm Hg. Then it means interstitial pressure exerting 10 and nephric filtrate exerting 10, and as well here what we have this osmotic pressure 30 mm Hg. Now this one, it means osmotic pressure, nephric filtrate pressure, interstitial pressure. These three combinedly opposing the blood pressure. It means we should add that 30 plus 10 plus 10, then it is a 50. This 50 gives opposite action to this 60 mm Hg. Now here we have a 60. This 60 minus 50. Then here whatever we got, it's a 10 mm Hg. With that 10 mm Hg, the blood it enters into the glomerulus. That is the thing here we have to remember. And now at what blood pressure the blood entered into the glomerulus is a 10 mm Hg, because this 50 mm Hg it's contributed by the osmotic pressure and as well interstitial pressure and as well nephric filtrate, and then it was entered. Then after entering, what's going to be happen? See, after entering this, what's going to be happen? Now remember the pressure, that's it. And here, let us see. Now it was entered. As of we know, this is an afferent arteriole. Arterioles opens into the capillaries, those are here present. And here, this area, it's made up of simple squamous epithelium. And this is made up of blood vessels as of we know, endothelium. And then endothelium of blood capillaries, that's blood vessels, and as well whatever we have is a simple squamous epithelium. Epithelium of Bowman's capsule. And now this is the parts. Now if you see clearly, this is also squamous epithelium, this is also squamous. Then these two have the both have the whatever we have here membrane, it was present. That membrane we called as a basement membrane. Basement membrane. Basement membrane is present in between these two. How it was present? Let us see once. It is seen here. It is an endothelium of blood vessels. And here we have a basement membrane at the middle. And here we have a squamous epithelium. And now the blood, whatever it came, it passing in this way. Whenever the blood passes here, the somewhat amounts of the nutrients it will be get inside here. and then this pores, whatever it was present here in between the endothelium, this is an endothelium, and this is a squamous epithelium. This squamous epithelium belongs to Bowman's capsule. This one belongs to the blood vessel, and the gaps between this endothelium we call here as a fenestrae. And the gaps between two squamous epithelium here, this one we are calling as a podocytes. Very important point, podocytes. Podocytes are the cells which are present here is on the squamous epithelium. That one can absorb the nutrients which are transferring in this, which are traveling in this direction, the blood, from the blood through the fenestrae. This vessels through that fenestrae, this all the blood nutrients and all it gets in, and then after in this direction it will enter into this region. It means this is what we are talking. Here all the nutrients and the somewhat plasma and all it entered into here. This is the place it was entered into the Bowman's capsule. Till now whatever we discussed, that is what we call as a ultrafiltration. And through the ultrafiltration, whatever it gets inside here, this is what we call as a nephric filtrate. We already discussed it. We should not call it as a urine at the starting, but this nephric filtrate has another name, that is what we call as a primary urine. Hope you understood the first step of urine formation, that is the ultrafiltration. In ultrafiltration, there it was happening in the Bowman's capsule, and here the simple absorption is happening. The simple logic here, if absorption is happening in that sense, it should be a simple epithelial tissue. That's squamous epithelial tissues required. Why? Because the reason that one only can able to be absorbed, because it is a very thinner epithelium. That's the reason it was present here, and also podocytes also helping it to absorb, and the pressure which are coming here with the 60 mmHg, that pressure it was got opposite action by the nephric filtrate contributes 10 and interstitial fluid contributes 10, and this glomerulus it contributes the 30 mmHg. Then this total 50 mmHg gives opposite action to this 60, and with the 10 mmHg pressure the blood entered into this region. Okay, that's all. And now we have to understand the simple thing here. The blood vessel, whenever it was coming out here, this is what we call as a afferent atrium. If you see the lumen of this afferent atrium, it is wider than afferent atrium. Very important point to understand. Afferent atrium is wider than efferent atrium. Then if it's wider, in this one all the blood and nutrients and all the things can able to be travel because it have the wider lumen. But if you see here, this one it have a very smaller one. If it have smaller one, it can't able to allow all the nutrients which are bigger. Okay, so that is the reason this one can't allow all the nutrients. Then which are present here, that is what we call as a nephric filtrate, also called as primary urine. It means in this traveling, the blood what is take along with that, it took here and it left somewhat the nutrients here, and then the remaining it was transferred in this direction with the afferent atrium. Then see here. Okay, then now let us see what is the composition of primary urine to understand the next step. Now let us see the next one. In our primary urine, what is primary urine? Or nephric filtrate. What it contains? See what blood contains. Blood contains plasma. Here, to the nephric filtrate, in the nephric filtrate, plasma is present. Okay, in the nephric filtrate, plasma is present. Along with plasma, the full of plasma was present. That's why we use it to call it's isotonic to plasma. Nephric filtrate is isotonic to plasma, and here, whatever the plasma it contains, but usually plasma have the proteins, but here that plasma minus proteins. It won't have any of the proteins. Now if we see what contains in afferent arteriole, okay, in afferent arteriole they have the blood cells plus proteins, and we have to see, remember here, when the blood came here at that time, plasma came into here, plasma came in this direction here, and the remaining what it contains, the blood cells along with the proteins, the two entered into the afferent arteriole. That's it. It means plasma in that sense, that's nothing but entire water and as well the somewhat inorganic molecules, and those all it was present here except proteins. That is what the composition of nephric filtrate. Now by using this data, let us understand the next step. Now, the nephric filtrate, whatever we have, that nephric filtrate, it was getting entered into the next place what it was present here. That nephric filtrate came here, the nephric filtrate, it came here, and then it enters here. This is an area we call it as a PCT. And here, we have to remember one thing. Per minute, in us, 1100 to 1200 ml of blood, it was getting entered into this afferent arteriole, and then it was crossing the afferent arteriole. It means per 1 minute this much of the blood it got filtering here, and as well in that blood apart from that, in this much of the blood it came here, in that sense how much of the plasma it was crossing here. Okay, how much of the plasma in that sense we have to remember here, it is 650 ml. In that 650 ml, how much amount of the particularly plasma it was getting into this glomerular film, into this Bowman's capsule, that one is 125 ml. This is what we call as a glomerular filtration rate, GFR, glomerular filtration rate. This glomerular filtration rate, it is 125 ml. It is the how much amount of the nephric filtrate it was forming per minute. Okay, if it's per a day, it is 180 ml. And now this GFR it should be constant. Okay, and as well it maintains a mechanism that one will discuss in a regulation of the kidney. Okay, and here it have a 125 ml, it was a GFR, what we have 180 ml per day. Very very important MCQs these both. Okay, that's all. And now this is what about the glomerular filtration. And now after the glomerular filtration, we are going to discuss about the next concept, the absorption. Where the absorption it was happening after the glomerular filtration, what we have. Here we are going to discuss about the next step, tubular reabsorption. What is tubular reabsorption? Okay, what is tubular reabsorption here? Remember the cheat code, reabsorption or absorption means from nephron tubule to blood vessel. That is what absorption. And secretion means from blood vessel to nephron. That's it. And here we are going to see reabsorption. Reabsorption means from where to where it is. From tubular parts of kidney. 2. From tubular parts of kidney to, from here to, it was going to get into the blood vessels. So that is what we call as a tubular reabsorption. And here in a tubular reabsorption, it was getting any of the thing, if it was getting entered from the tubular parts of the kidney to the blood vessel, that is what we call as a tubular reabsorption. And see, in tubular reabsorption, what the thing we have to remember, first we have to know what is PCT. This PCT, it was made up of, how this PCT is made? It was made up of a brush bordered cuboidal epithelial tissue. And remember, here cuboidal given, in somewhere it was columnar also. Okay, it was somewhere it was columnar also. That's why we call it as a tubular and as a columnar. Keep that in mind. And as well, why brush border? Because of the absorption. Because of the absorption. Then what it was going to be absorbed here? The things which are present there, we have to remember. Okay, we already discussed what is present here in the, that is nothing but plasma. Okay, what it includes? It includes glucose, it includes lipids, it includes a lot of amino acids. These are all the things it included there. What does it means? It means it have a lot of things are there which are required to our body. That is the reason it was reabsorbing from the tubular part of kidney to blood vessel. If it was getting into the blood vessel in that sense, it was utilizing for our metabolism. So that is the reason it was going to be reabsorbed. Then what it includes? Nothing but glucose, amino acids, and as well sodium, and as well few kind of lipids. But these are the things it reabsorbed. Reabsorbed means, as of we discussed it, from nephron to blood vessels. That's it. Then those are entering. But how they are entering? It have they are entering through the active process. Then if it's an active process, it required a mitochondria. That all mitochondria, it was present in a PCT. Then for reabsorption, it required an epithelium. That's why it have the brush bordered epithelium, cuboidal epithelium, and also it have the mitochondria. It releases the ATP, and by using that ATP molecule, it undergoes this active process, and then it reabsorbs these all the nutrients which are required to our body. That's it. And if you think there is an only active process, passive also there. Passive process also there, but through the passive process which are getting entered here, nothing but water through the simple diffusions and as well chloride ions. These two are getting entered. That's all. And here we see what it was happening in tubular reabsorption. Here from kidney to blood vessel it was transferring, and what to transfer, which it have the structure, it includes brush bordered epithelium. And that's why absorption is happening, which are absorbing glucose, amino acids, sodium ions through the active process, which are through the passive process, water and as well chloride ions. And for active process, what is required? ATP is required. That is the reason it have the mitochondria. That's all. And next, this one also includes a very important function. That function is a selective secretion. Selective secretion. What is selective secretion? What is selective secretion in that sense? Selective secretion means selectively secreting. Okay? Selectively secreting in that sense, secretion means we already discussed from blood vessel to nephron, which are transporting here, which are not completely, which are not required for our body. That one is transporting from blood vessel to the nephron here. Then, which are not required, those are nothing but drugs, a kind of drugs, and as well a kind of waste. These are the things it was separated into the nephron part. That is nothing but in a DCT. That is nothing but in a PCT point of view. And then, which are selectively secreting here? The selectively secreting one is potassium, and as well along with the somewhat let's see the somewhat like H2O. The somewhat, these are the things. It was instead of H2O, we can say H plus ions. H plus ions and as well potassium ions. These both are selectively secreting. For what purpose they are selectively secreting here? To reabsorb the HCO3 minus. For what purpose HCO3 minus have to be retake? Why? Because we already seen in the respiration concept, it was reabsorbing for the purpose of transport of the carbon dioxide. We know that bicarbonate ion, in the form of bicarbonate ion, carbon dioxide transport, it will be 70%. So for that reason, this bicarbonate ion should be taken. To take that bicarbonate ion, selectively it was secreting this H plus and as well this potassium ion also. And these two, it was happening here. And now let us see here. This one, what we have to remember here, this process it was happening here. For that reason, about this all tubular reabsorption, it was happening in PCT only. Then if it was happening in a PCT in that sense, then what purpose these all are happening here? For to reabsorb this HCO3 minus and as well to maintain the pH and ionic balance. To maintain pH and ionic balance in PCT. This is the main function of this whatever the selective secretion and as well absorption of this bicarbonate ion. This is the main theme of this selective secretion, and then this, by doing that activity, it maintains the pH and ionic balance here. That's all. That is what we call as a tubular reabsorption. And the next one. And we have a few more steps. That is what in it happens in a loop of Henle. What happens in loop of Henle? In loop of Henle, there is a thing what's going to be happen here. The absorption and the secretions are minimal. Nothing is going to be happen. The remaining things, it comes here, and here we have the two things. This is a descending limb. It's an ascending limb. It's an ascending limb. Descending and ascending limbs we have. And the descending limb which we have, that descending limb, it was transported. What it will do? Okay, it was permeable to water and as well impermeable to electrolytes and as well. And this is an opposite action, ascending limb. This ascending limb, opposite action, permeable to electrolytes, impermeable to water. It means these both are maintaining the opposite action. By these opposite actions, they will maintain the counter current mechanism. That is what their duty of that. And as well, the next one, by maintaining this activity, it maintains the proper concentrations in the medullary interstitial fluid. That is what the activity of this loop of Henle by having the two limbs. That is what the function of it. And at the end, it will go to the DCT, and at the DCT also same functions it will happen, and as well it maintains the same concentrations and all, and then finally it comes to the collecting duct, and then finally this collecting duct, it opens into that pelvis region, and then it was transferred to the urinary bladders, and then it will get into the by the micturition process, that urine it will be expelled out. That is what the urine formation, and as well the entire contents what about this structure of the nephron and this urine formation. This all